In replication, a molecule of DNA will serve as a template for two new identical daughter strands. The bonds of a DNA molecule will break, with each strand acting as a template for a new complementary strand produced with the help of DNA polymerase. This results in two molecules of DNA. In transcription, the information in a strand of DNA is copied into a new molecule of mRNA using RNA polymerase. Matching nucleotide bases are paired to the original DNA strand. This sequence of bases, called mRNA, will serve as instructions for protein synthesis during translation. Translation is the process by which a protein is synthesized from the information contained in a molecule of mRNA. The main components of translation are mRNA, a ribosome containing the small and large ribosomal subunits, as well as the E, P, and A sites, amino acids, tRNA, codons, and anticodons. The mRNA, tRNA, codons, and anticodons were made from construction paper, ribosome from a shoebox, and amino acids from plastic eggs. The amino acids are methionine, valine, alanine, and leucine. Translation begins with initiation in the nucleus. In initiation, the small ribosomal subunit will attach to the 5' end of the mRNA strand in search of the start codon, AUG. This is followed by a tRNA molecule carrying methionine binding to the start codon. The process ends with the large ribosomal subunit binding to the site. The next process, elongation, begins when the tRNA molecule complementary to the start codon bonds to the codon and moves to the P site. Another tRNA molecule, based on the complementary codon, will move to the A site. The amino acid in the P site will form a peptide bond with the amino acid in the A site, pairing them together. The tRNA molecule in the P site moves to the E site, where it exits the ribosome, and the tRNA molecule in the A site moves to the P site. Another tRNA molecule enters the A site, and the two amino acids bond to the amino acid in the A site. This process continues until a stop codon is in the A site. A stop codon can be UAG, UAA, or UGA, and in this case, it is UAG. This causes a whole complex to dissociate, leaving the newly formed protein.